Good evening, everyone. Myself, Dr. Kumari Sunita, Assistant Professor, Deen Dayal Upadhyay, Gorakhpur University, Gorakhpur. Today, we will study about the physiological effect of gibberellin. Gibberellin was discovered for the first time by a Japanese scientist Kurosawa in 1930 in Japan and Formosa. It was observed in rice field that a few plants were distinctly taller, seedless, and pale in color in comparison to normal rice plants. It was a disease called foolish seedling disease or Bacan disease of rice. It was caused by a fungus Gibberella fugicori, that is also called as Fusarium moniliforme. One of the most prominent effects of gibberellin is their promotion of stem elongation. This effect is particularly dramatic in dwarf mutant, where the application of gibberellins can restore normal growth. Gibberellin can also stimulate both cell division and cell elongation in the stem, leading to increased internode length. This effect is achieved through the activation of genes involved in cell wall loosening, such as those encoding expansins and gyroglucan endotransglucosylase hydrolases, that is also called as XTHS. Gibberellins overcome seed tuber and bud dormancy by countering the inhibitory effect of abscisic acid, a hormone that promotes dormancy. The application of gibberellins can trigger premature sprouting in potato tubers and tree buds during the winter month. In this slide, we can see the effect of gibberellin on germination. Gibberellin promote germination and it stimulate the production of alpha amylase, which hydrolyzes stored starch into simple sugar, providing energy for seedling growth. Digestive food present in seed is utilized in seed germination. It promotes seed germination and antagonistic to ABA because ABA, that is also called as abscisic acid, we all know, act as a main factor for dormancy in plant. Period of seed dormancy is about 10 to 12 years and gibberellic acid promotes quick germination of seeds. There are two types of seeds. First one is photoblastic seed and second one is non-photoblastic seed. Photoblastic seed, the seed germination in presence of light, for example barley, and in non-photoblastic seed that can germinate either in presence or absence of light, for example mustard, pea and gram. And gibberellic acid promotes germination of photoplastic seed even in complete darkness. Flowering occurs due to florism hormone and gibberellic acid induce formation of florism hormone, hence promote flowering in long day plant, even in short day condition, or help transition from vegetative growth to reproductive growth. Gibberellin stimulate the growth and development of pollen grains. Likewise, gibberellin also promote the formation of fruit and their development, and it is also used for the production of parthenocarpic fruit, that is also called as seedless fruit, that the fruit from without fertilization. Leaf expansion. Gibberellic acid also promotes cell division, increase expression of aquaporins, leading to high pressure, which stretches the leaf cells and promote the expression of expansion proteins, which help loosen the rigid cell wall, facilitating expansion in developing leaves, contributing to leaf size. In some species, gibberellin can affect leaf shape, such as promoting the formation of divided leaves in certain aquatic plants. The transition from juvenile to adult phases in woody plant is another process regulated by gibberellin. High level of gibberellins are associated with the juvenile phase, and a decrease in gibberellins level or sensitivity often accompanies the transition to the adult reproductive phase. Gibberellin can also delay the aging of flowers, which allows for longer period of flowering and potentially longer fruit development period. Vernalization works by suppression of expression of certain genes that inhibit flowering. For example, FLC gene in Arabidopsis. Cold temperature usually downregulate the FLC gene, allowing flowering to occur. Gibberellin can interfere with the vernalization process by either directly or indirectly reducing the level of FLC expression, or by promoting the expression of flowering gene that bypass the cold requirement. Gibberellin interact with various flowering related pathways, including those that involve floral induction genes. In some plants, gibberellin promote the expression of gene like SOC1 or LFY, which are important for flowering, thereby facilitating flowers development without the need of vernalization. Gibberellin play a role in enhancing metabolic activities within the plant. They can promote the production of protein and enzymes needed for various physiological processes. This includes activation of ribosomal RNA synthesis, leading to more protein production, which is vital for growth. This is all about physiological effect of gibberellin. Thank you so much. Namaskar.